Hello students, a very good morning to one and all. Welcome to the next lecture of online theory class series of subject Novel Drug Delivery System. I am Mrs. Aarti Majumdar, Associate Professor from the Department of Pharmaceutics, Smriti College of Pharmaceutical Education, Indore. Dear students, in continuation of our discussion over nanotechnology and targeted drug delivery system, so far we have discussed about basic concept of nanotechnology and targeted drug delivery system, including what are targeted drug delivery system, what is the objective behind the development of targeted drug delivery system, what is the principle and rationale of drug targeting, what are the different types of carrier used in the formulation of targeted drug delivery system, and what are the various approaches used for targeting drug candidates to the particular site of action. So in continuation of discussion in various approaches, what are the various types of approaches? So far we have discussed about passive targeting, inverse targeting, active targeting, including organ compartmentalization, then cellular targeting and intracellular targeting. In today's session, we are going to discuss about what is dual targeting, double targeting and combination targeting. In today's session, we will also discuss about what are the various advantages and limitations of targeted drug delivery system. Now, starting with dual targeting. In this approach, dual targeting, such carriers which have their intrinsic therapeutic effect, they have their own therapeutic effect or pharmacological effect, such type of carriers are used. For example, a carrier which is having antiviral effect. If such carrier are loaded with an active antiviral drug, then the antiviral activity of drug carrier system, it is fortified, it is found to be increased, and also the synergistic effect is observed because with the help of antiviral activity of both carrier and drug. In this type of targeting, and the most important advantage is that the viral replication, it is attacked at multiple points. This helps to minimize the development of resistant strain, viral strain. So this is dual targeting, in which carrier which are having their own pharmacological action, own therapeutic effect, they are utilized. Now, double targeting. Double targeting, it is a targeting technique, targeting approach in which is combined with other methodologies which are other than active and passive drug targeting, such as in double targeting, spatial delivery, control over spatial delivery, it is combined with the controlled delivery in terms of temporal controlled delivery. I must remind you, we have studied what is spatial drug delivery and what is temporal drug delivery in our unit controlled drug delivery system. So, spatial drug delivery, it refers to the control of delivery of drug with respect to the particular site of drug release. In spatial delivery, control over the drug release is done with respect to the particular site. The drug is released in the body on reaching to the particular site, whereas in temporal drug delivery, it refers to the controlling the release of drug with respect to the rate of drug release, or maybe with respect to the time of drug release. So, in double targeting approach, both these methodologies, both these approaches, that is spatial control as well as temporal control, can be, they are both combined and have their advantages. For example, temporal drug delivery, it can be achieved with many ways we have studied in our chapter, in our unit controlled drug delivery system. They can be sustained release formulation, or the drug release can be stimuli activity chemically activity systems are there. For example, glucose responsive system, enzyme activated system. We have studied all these types of temporal control system or control release system in detail. So when such approach they are utilized, and when the carriers are used which direct the drug spatially, which direct the drug to reach to the particular site where, and the drug is also released. Drug is controlled temporarily. Drug release is controlled temporarily so that the drug release or activation takes place upon reaching to the site of action. So this will further enhance the therapeutic effect of drug. Now, combination targeting. This combination targeting approach is utilized in the targeting of proteins and peptides. Especially in this combination targeting, it approaches these targeting systems. They are equipped with carriers, polymers, or homing devices, which may be antibodies or other polymers, proteins. These homing devices, which are of molecular specificity, that could provide a direct approach to the target site. So what happened? Initially, proteins and peptides which are to be targeted to a particular site, so they have to be modified with the help of either natural polymer, for example polysaccharides, or maybe with the help of synthetic polymers, for example polyethylene glycols. So after modification, this modification alters the physical characteristics of these proteins and peptides. After modification, vectorization of these modified proteins and peptide is done with the help of various vesicular or microparticulate colloidal carriers. So this combination, because of this combination, it is called as combination targeting. It further increases the therapeutic effect of therapeutic bioavailability of targeted drug. Now students, let's discuss about what are the various advantages of targeted drug delivery system. So first and most important advantage is that selective targeting to the disease cell, so that it spares healthier cell from over drug exposure, and reduced side effect will be there. Now, avoidance of hepatic first pass metabolism. If the drug will be released at the site of action, then hepatic first pass metabolism will be minimized. Then increased patient compliance, and low dose is required for producing the desired pharmacological effect. And because of this low dose and selectivity, tissue selectivity, so it results in decreased side effects. Apart from having so many advantages, these targeted drug delivery system, they are also having certain limitations, which may be rapid clearance of targeted drug delivery system, especially antibody targeted drug delivery system. They are rapidly cleared from the circulation, from the body. Then sometimes immune reactions are observed against intravenously administered carriers. Then target tissue heterogeneity. Then another limitation is insufficient localization in tumor cells. Sometimes tumor cells are having lower permeability as compared to the normal cells. So because of that, insufficient localization of drug is observed, because tumor cells are having somewhat different vascularity. Then diffusion and redistribution of drug is another disadvantage while dealing with targeted drug delivery system. So dear students, this is all about targeted drug delivery system. In our next lecture, we will start our discussion over various carriers such as liposomes, nanosomes, monoclonal antibody, etc. So we will study in detail about what are these carriers, how they are formulated and evaluated, and what are the various applications of these carriers in pharmaceuticals. Thank you.